BBIU Edu, from lab bench to FDA approval, why Module 3 defines the fate of every new medicine, September 19th. When people hear about drug development, they think of clinical trials, patients receiving an experimental therapy, results published in journals, and sometimes even headlines about breakthroughs. But hidden behind those clinical milestones lies a quieter, less glamorous document that can make or break the entire project. Module 3 of the CTD, or Common Technical Document. Module 3, known as the Quality Dossier, is where science is forced to prove it can become industry. It is the bridge between discovery and the pharmacy shelf, and the lens through which regulators decide if a medicine can be trusted to reach the public. Why Module 3 matters, the backbone of quality. If Module 5 of the CTD shows what a drug does in humans, Module 3 shows whether it can be reliably reproduced. A clinical trial may prove efficacy and safety once, but Module 3 asks, can this drug be made the same way every time at industrial scale, the anchor of trust? Regulators like the FDA, EMA, MFDS, and PMDA lean heavily on Module 3. It assures them that every vial, tablet, or injection leaving the factory is consistent, stable, and safe. The gatekeeper. Even the most impressive clinical data cannot pass into approval without a robust Module 3. It is the gatekeeper between science and commercialization. What happens when compliance is weak? Regulatory refusals and delays. The FDA can refuse to review an application, issuing a refuse to file notice, or send a complete response letter if Module 3 is incomplete, inconsistent, or poorly validated. In Europe, the EMA can issue major objections that delay or even withdraw an application. Market disruptions. Inadequate stability data has forced companies to pull applications mid-review, wasting years of work. Financial losses. One deficiency in Module 3 can result in hundreds of millions in lost revenue due to launch delays. Patient risk. Weaknesses in Module 3 can lead to drug recalls, shortages, unsafe products on the market, and erosion of public trust. From lab scale to mass production. Every new drug starts in a lab flask, but scaling from milligrams to metric tons is the true test of Module 3. Lab scale production. Context. Academic labs or small biotech R&D. Characteristics. Milligram to gram quantities, manual purification, limited analysis. Risks. Lot-to-lot variability, fragile processes, and non-reproducibility. Relevance in Module 3. This phase supports early structural description and initial synthesis documentation, but it is not yet sufficient for commercialization. Mass production, pilot to commercial scale. As a drug advances into Phase 2 and 3 trials, large-scale production becomes essential. Characteristics include kilogram to ton scale manufacturing under GMP conditions, validated processes, strict controls for critical quality attributes, impurity monitoring, and robust stability programs. Critical sections in Module 3 include 3.2.2 Detailed Manufacturing Description, 3.3.3 Product Validation, and 3.2.8 Stability Testing. Risks at this stage include new impurities not seen at lab scale, instability before expiry, and inconsistency between clinical and commercial lots. The transition, where companies fail. Small companies often master lab synthesis but stumble at mass production. History provides notable examples. Ritonavir, where a new crystalline form appeared during scale-up, disrupting global supply, and NDMA impurities in sartans in 2018, where industrial optimizations unintentionally produced carcinogenic byproducts, triggering worldwide recalls. The lesson: lab scale proves the scientific concept. Mass production proves commercial and regulatory viability. Module 3 documents the bridge. Drug material used in clinical trials, lab versus mass scale. Phase 1 typically uses lab or small pilot batches. Only grams are needed, and regulators accept this if material identity and purity are well characterized. Phase 2 involves larger batches, often in kilos. These may still be pilot scale, but regulators expect transparency and justification. Phase 3 is the turning point. Clinical material must be manufactured using a process nearly identical to the commercial one, with validated stability and comparability studies. For commercial launch, only GMP-certified, validated commercial processes are acceptable. Any deviation from Phase 3 material requires regulatory supplements or variations. Risk of non-compliance. If clinical trials use lab-made drug that differs from commercial material, regulators may require new studies or even repetition of Phase 3. This has delayed several biosimilars in Europe. Process improvements, yield and purity. While improving yield or purity sounds positive, regulators see risk. Concerns include new impurity profiles or crystalline forms. Comparability studies are needed to prove equivalence with clinical material. Regulators favor consistency over unvalidated improvements. Outcomes. If comparability is proven, the change is accepted as a CMC supplement or EMA Type 2 variation. If not, new bridging trials may be required, costing years. Examples. In biologics, switching fermenters can improve purity but alter glycosylation. In small molecules, new solvents may reduce impurities but introduce trace contaminants. NDMA in sartans is a cautionary tale where yield optimizations introduced carcinogens, prompting global recalls. Strategic lesson: every process improvement is a regulatory event. More pure does not mean automatically acceptable. Bridge tests: original versus improved drug. When manufacturing processes change, regulators require proof that the new version of a drug is equivalent to the original. The bridge package typically includes analytical comparisons such as impurity profiles, crystallinity, solubility, and potency. It may also require non-clinical toxicology for any new impurities and clinical studies if necessary, like pharmacokinetics or immunogenicity. Terminology varies. The FDA uses a comparability protocol via CMC supplements. The EMA calls it a comparability exercise via Type 2 variation. ICH-Q5E is the reference for biologics, but the concept applies widely. Skipping a bridge test can lead regulators to consider the new material a different drug, requiring clinical trial repetition or even rejection. One example: a generics company improved drug purity from 95% to 99.5%, but introduced a 0.2% impurity. The EMA required identification, rodent toxicology, and a clinical bridge before approval. How the FDA inspects Module 3 in practice. Approval goes beyond documents. FDA inspectors visit manufacturing sites to confirm Module 3 is implemented in reality. They check SOPs, standard operating procedures, for accuracy and compliance. Batch records are reviewed to compare the master recipe to actual execution. Sampling and retention practices must be traceable. Certificates of analysis from suppliers must be independently verified. Impurity limits are reviewed, with CAPA enforcement for any breach. Stability data must include at least 12 months real-time and 6 months accelerated results. Full traceability is required for all raw materials, excipients, and containers, including temperature logs. Packaging must protect the drug and comply with labeling laws. SOPs are central. They prove that Module 3 is being followed. Weak or ignored SOPs lead to Form 483 observations or even warning letters, delaying or blocking approval. Final takeaway. For the public, clinical trial results make headlines. For regulators, Module 3 is the foundation of trust. It ensures a medicine can be made safely, consistently, and at scale. Module 3 connects discovery to industry, science to commerce, and patients to reliable treatment. It is not just paperwork, it is the living blueprint of a drug's quality, verified by factory inspections, enforced through SOPs, and grounded in global standards. Without Module 3, no new therapy reaches the market, no matter how promising its science. Annex 1. Shelf life, aging tests, and post-approval quality management. 1. Shelf life determination. Shelf life is the time during which a drug product is guaranteed to remain within approved specifications. It is established through long-term stability data, typically 12 months or more, and 6 months of accelerated testing. If long-term data are incomplete, provisional shelf life may be granted with a commitment to submit updates. Regulators see shelf life as a scientific conclusion, not a marketing tool. 2. Aging tests or stability studies. These simulate real-time and stress conditions. Real-time aging involves 25 degrees and 60% RH for 24 to 36 months. Accelerated aging uses 40-degree cells and 75% RH for 6 months. Intermediate conditions and stress testing, like exposure to light or oxygen, help reveal degradation risks. Approval requires a minimum of 12 months real-time and 6 months accelerated data. Post-approval, at least one commercial batch per year must undergo stability testing. 3. Documentation of lots and sampling. Batch records must detail each lot's production. Retention samples are stored for the full shelf life, plus 1 year. Sampling SOPs define how stability samples are drawn. FDA inspectors verify that samples exist, are intact, and match what's declared in Module 3. Certificates of analysis, or COAs. A supplier COA accompanies raw materials, but the manufacturer must always test at least for identity. Other tests may be waived only after supplier qualification. The FDA expects companies to verify COAs. Relying solely on a supplier's report is non-compliant. Impurity limits and CAPA. Impurity limits are set by the sponsor based on ICH Q3A or Q3B guidelines and toxicological data, and are then approved by regulators. If a batch exceeds limits, it's recorded as an out-of-specification event and triggers a CAPA plan, corrective and preventive action. FDA inspections focus on seeing not just the correction, but the documented prevention strategy. Post-approval lifecycle changes. Shelf life can be extended with new stability data. Changes in manufacturing sites, equipment, or raw material suppliers all require regulatory notification or approval, such as a supplemental NDA or EMA Type 2 variation. ICH Q12 encourages companies to plan for such changes through a post-approval change management protocol, or PACMP. Packaging and shelf life. The container closure system must protect against moisture, light, oxygen, and mechanical stress. Labels and inserts must include the product name, strength, dosage form, batch number, expiry date, and storage instructions. FDA checks that the packaging used in stability tests matches what is used commercially. Educational takeaway. The shelf life printed on every drug package is not arbitrary. It is the final outcome of a rigorous process: stability studies, batch documentation, verified COAs, defined impurity limits, CAPA systems, and regulatory approval in Module 3. These systems together ensure that a drug remains safe and effective until its expiration date. Annex 2. Real-world examples of Module 3 in action. 1. Ritonavir. During scale-up, a new crystalline form of ritonavir emerged that was more stable but less soluble, making the drug ineffective at standard doses. The result was a global withdrawal, reformulation, and major losses. The lesson: polymorphism must be fully characterized and stability must account for crystal form changes. 2. Heparin contamination crisis. In 2008, heparin contaminated with oversulfated chondroitin sulfate caused severe allergic reactions and patient deaths. Routine quality checks missed it. The lesson: supplier COAs are not enough. Identity testing and qualification are essential. 3. NDMA and sartans. In 2018 and 2019, process optimizations at plants in China and India introduced carcinogenic nitrosamine impurities. Global recalls followed. The lesson: every process change must be validated and impurity profiles reassessed. 4. Biologics glycosylation shifts. Switching cell lines or fermenters changed glycosylation patterns, delaying approvals. The lesson: even subtle changes in biologics require comparability studies. 5. Generics case. A generics firm improved purity from 95% to 99.5%, but a new 0.2% impurity appeared. EMA required toxicology and bioequivalence bridging. The lesson: different equals different until proven otherwise. 6. Stability failures. Several small molecule drugs were withdrawn due to degradation revealed in stability testing. The lesson: shelf life must be earned through validated aging studies. Educational takeaway. Module 3 isn't paperwork. It's the frontline defense against unsafe or unstable medicines. Quality failures, even after clinical success, can erase years of work and billions in investment.
Welcome to the BioPharma Business Intelligence Unit, where symbolic integrity meets strategic clarity. At BBIU, we go beyond the headlines to decode the structural forces behind global biopharma, AI, and economic realignment. If you value precision, verification, and symbolic depth, this channel is for you. Subscribe now to access our weekly briefings, dossiers, and intelligence reports, and consider joining our institutional tier to unlock early warning systems, custom metrics, and exclusive strategic insight. You're not just listening, you're entering the future of epistemic intelligence. www.biopharmabusinessintelligenceunit.com